Hello, and a very warm welcome to everyone joining us for today's Select Science webinar titled Bispecific Antibody Overview and Production with CrossMab Technology. My name is Lawrence Howes and I'll be moderating today's presentation. I'm delighted to be joined by our guest speaker, Dr. Sai Wang, Protein Senior Scientist at GenScript Biotech Singapore. This webinar will discuss the advantages of bispecific antibodies, their mechanisms of action, and the development history of bispecific antibody platforms. She will also help attendees gain an understanding of the CrossMab production method and its significance in the field of antibody engineering. Following the presentation, we'll have time for a question and answer session. If you have any questions throughout the webinar, please feel free to submit these in the box to the right of your screen. So without further delay, I'd like to hand over to our speaker. I'd like to thank them again for presenting for us today.
Thank you very much for the introduction. Hello, everyone. Welcome to the webinar. The topic today is bispecific antibody overview and production with the CrossMab technology. Here is a preview of today's talk. I will first give you a brief introduction of bispecific antibody, which will then be followed by examples of different mechanisms of action, followed by the development of history of bispecific antibody platforms and commercialize the bispecific antibody. In the later session, I will be focusing on CrossMab technology and showcasing production of 2 CrossMab bispecific antibodies by GenScript mammalian recombinant protein production platform. Last but not the least, I will give you a quick introduction of GenScript virtual mammalian platform. Typical monoclonal antibodies target one epitope. FDA approved the first monoclonal antibody, OKT3, by commercial name Orthoclone, in 1986 to help prevent rejection in organ transplantation. A bispecific antibody is an artificial protein that has 2 distinct binding domains that can bind to 2 antigens or 2 epitopes of the same antigen simultaneously. Over the past 2 decades, bispecific antibody development has been revolutionized due to genetic engineering approaches that enable a wide variety of molecular structures that offer different advantages. By targeting 2 antigens or epitopes, they can cause multiple physiological or anti-tumor responses, which may be independent or connected. These treatments may act like a cocktail of 2 monoclonal antibodies, but drug developers need to manufacture only 1 molecule, and the patients may need only 1 antibody treatment. Plus, their synergistic or cooperative features may produce more significant treatment effects. Currently, most bispecific antibodies in development aim to treat cancer, but others are focused on chronic inflammatory, autoimmune, and neurodegenerative diseases, vascular, ocular, and hematologic disorders and infections. Now let's take a look at examples of mechanism of action of bispecific antibodies. The majority of bispecific antibodies in clinical trials are CD3 T-cell engager. These bispecific antibodies bind to CD3 except on the surface of T cells and to a tumor-associated antigen on the tumor cell surface. When both CD3 and the TAA are engaged, the proximity of the T cell and the tumor cell results in the formation of an immune synapse, elimination of the T cell, and redirection of cytotoxicity against the T cell, against the tumor cell. Bridging 2 different receptors on one cell is another mechanism in which the cross-linking of specific cell surface receptors leads to their inactivation, for example, to reduce tumor growth in cancer applications, or activation, for example, to conditionally activate a growth factor receptor for the treatment of diabetes. Bispecific antibodies may be designed for the precise positioning of an enzyme and an substrate as a cofactor mimetic, such as this bispecific antibody to replace a critical clotting factor in the treatment of hemophilia. In all these discussed mechanisms, binding of the bispecific moieties occurs simultaneously. There are also mechanisms of action in which binding of the bispecific moieties occurs sequentially. Piggybacking allows the translocation of an active binding arm to an otherwise inaccessible compartment, such as central nervous disease targets protected by the blood-brain barrier, or bacterial or viral antigens with a role in endosomal escape. To date, more than 20 different commercialized technology platforms are available for bispecific antibody creation and development. The original concept of bispecific antibodies was proposed by Nisonov and his collaborators in the 1960s, including the first idea of antibody architecture and the first fragment-based format. In 1975, the problem of producing pure antibodies was solved by the creation of hybridoma technology, and the new era of monoclonal antibodies came. In 1983, hybridoma technology was created. In 1988, the single-chain variable fragment, SCFV, was invented to minimize the refolding problem and the chain association issue. In 1995, the first solution to light chain association issue was developed through species-restricted light chain pairing. In 1996, the bispecific antibodies became more developed when the knobs into holes technology emerged. Heavy chain association issue was minimized through the use of complementary heavy chains. In 1998, common light chain was first used to tackle the light chain pairing problem. In 1999, it was discovered that natural human IgG4 is bispecific. The following two highlights were in 2007, which are the discovery and the elucidation of bispecific human IgG4 Fab arm exchange process in vivo, and the invention of dual variable domain Ig symmetric format. In 2011, CrossMab technology using domain crossover was developed to solve light chain association issue. So I will further elaborate on CrossMab in the second part of this talk. In 2013, bispecific IgG1 was generated through Fab arm exchange of separately expressed antibodies. In 2014, orthogonal Fab interface was first discussed as a solution to light chain association issue. Currently, there are 13 bispecific antibodies approved worldwide. Among them, 11 were approved by FDA, as highlighted in blue. And as you can notice, majority of these bispecific antibodies, in total of 10, were approved in the last 2 years. So in the next section, I will be focusing on CrossMab bispecific antibody technology, which were applied in the development of Glofitamab and bsAb. As has been briefly introduced earlier, CrossMab technology was first described by Roche in 2011 as a novel approach enabling correct antibody light chain association with their respective heavy chain in bispecific antibodies. Since the original description, CrossMab technology has evolved in the past decade into one of the most mature and broadly applied technologies in the field, and dozens of bispecific antibodies based on CrossMab technology developed by Roche and others have entered clinical trials. The most advanced of these are Glofitamab and bsAb. Both have been approved by FDA and EMA in the past 2 years. The co-expression of 2 different heavy chains and 2 different light chains results in a complex mixture of 16 possible recombinations representing 10 different antibodies. Only one of these antibodies corresponds with the desired antibody, the maximal yield of which is only 12.5% in the mixture. 2 problems must be solved to produce the desired bispecific antibody exclusively. First, effective induction of heterodimerization of the 2 heavy chains. Second, discrimination between the 2 light chain heavy chain interactions. Correct heavy chain association resulting in a heterodimeric Fc can be enforced using knobs into holes technology by introducing a bulky tryptophan residue in one Fc fragment and forming a corresponding cavity on the other Fc fragment that can accommodate the tryptophan residue. More recently, multiple alternative approaches to enable correct heavy chain association have been described, such as relying on charge interactions. Although knob into hole technology was developed in the late 1990s, enabling correct light chain association remained a major problem, and the only approach to achieve this before CrossMab was invented relied on the use of common light chains for both specificities. However, the use of a common light chain requires the de novo identification of the corresponding antibody pairs, which can be challenging and time-consuming depending on the desired target, and restricts the availability and diversity of the antibodies that can be used. Thus, methods allowing the generation of bispecific antibodies from pre-existing antibody pairs were highly desired. CrossMab technology innovated a domain crossover strategy to enable correct light chain association in bispecific antibodies. Here goes the basic principle. The original heavy chain VHH1 domains were incorporated as the novel light chain, and the original VLCL domains were fused to the hinge region of the Fc fragment as the novel heavy chain. Correct light chain association can be enforced in this CrossMab Fab format. This format has also been described as Fabs in tandem RG format. Alternatively, only the VHVL or only the CH1CL domains can be exchanged in the CrossMab VHVL and the CrossMab CH1CL format. In the case of CH1CL crossover, no theoretical side product due to domain crossover is expected, and crystal structure analysis confirmed the structural integrity of the crossed Fab domain in the CrossMab CH1CL format. In the case of Fab crossover, 2 heavy chain and light chain based side products can be observed. However, the correct preferential formation of the CrossMab Fab can be fostered by relative overexpression of the respective light chains, so that the respective undesired monovalent and binding inactive side products do not form in significant amounts. Similarly, In the case of VH-VL crossover, a Benz-Jones-like side product based on VL-VL together with the CH1CL interaction can be observed. In order to avoid formation of this side product, natural charge pairs in the Fab were identified, and the respective orthogonal charge interactions were introduced into the non-exchanged antibody CH1CL domains. As a consequence, the undesired Benz-Jones-like side product does not form due to repulsive charge interactions, whereas the desired light chain pairs correctly in the non-crossed Fab due to attractive charge interactions. So in the following slides, I'll be showcasing production of two CrossMab bispecific antibodies by GenScript mammalian recombinant protein production platform. Glofitamab, which was sold under the brand name Columvi, is a bispecific CD20-directed CD3 T-cell engager developed by Roche. Glofitamab received the approval in 2023 for the treatment of adult patients with relapsed or refractory diffuse large B-cell lymphoma, DLBCL. Glofitamab has a 2-to-1 configuration with a bivalency towards CD20 and monovalency towards CD3, and works by recruiting T cells directly to the surface of cancerous B cells. Correct heavy chain association was enforced by knobs into host technology. Correct light chain association was enabled by CrossMab VH-VL domain crossover. And in order to avoid formation of Benz-Jones-like side product, orthogonal charge interactions were introduced into the non-exchanged antibody CH1CL domains. Antibody sequences were optimized by GenScript proprietary codon optimization algorithm. After an initial small-scale chain ratio screening, 1 liter scale-up production was done using turbocharged transient expression system. Clarified cell culture supernatant went through 2 steps of chromatographic purification using protein A affinity method, followed by size exclusion method. Purity of the target antibody was examined by SEC-HPLC and CE-SDS. The yield of purified antibody was 24 milligram per liter of cell culture. Purity was 95% indicated by CE-SDS and over 99% as indicated by SEC-HPLC. Endotoxin level was below 0.1 EU per mg of purified antibody. The antibody molecule from case study 2 takes the 1-to-1 CrossMab CH1CL format. Correct heavy chain association was enforced by knobs into host technology, and the correct light chain association was enabled by this CH1CL crossover. Antibody sequences were optimized by GenScript Zhu-Che codon optimization algorithm. First, an expression was done in 100 ml turbocharged transient expression system. Clarified cell culture supernatant went through 2 steps purification using protein A affinity chromatography and capture select kappa affinity chromatography. The yield of purified antibody was 189 milligram per liter of cell culture. Purity was over 95% by SDS page and over 95% by SEC-HPLC. Endotoxin level was below 0.1 EU per mg of purified antibody. So we are almost to the end of today's talk. A quick summary of what have been shared. First, bispecific antibodies and their mechanism of action were introduced, followed by a a chronological brief review of the development of bispecific antibody technology platforms and the overview of commercialized bispecific antibodies. We then moved on to the introduction of CrossMab technology in more details, followed by 2 case studies of CrossMab production by GenScript Mammalian platform. GenScript is providing proprietary TurboCHO platform, which offers high yield performance within a short turnaround time. And this cost-effective production system enables a small to large-scale production of antibodies with a high level of consistency, making it an excellent complement for target discovery and a pre-evaluation pipeline. And empowered by the strong R&D capability, TurboCHO platform provides unparalleled services for bispecific antibody production with a specialized format design, AI-driven codon optimization, optimized plasmid ratio and production process, and specialized multi-step purification method. GenScript has provided an updated bispecific antibody website for you to take a look at. If you scan the QR code here, you will see all the details. And the highlight is the interactive bispecific antibody table. The molecules in the table are classified by antibody fragment, symmetric, and asymmetric format. If you click on a pictogram, you will see more details of the molecule, namely the manufacturability of that format, recommended purification methods, typical yield, and purity. This comes to the end of my talk. Thank you for watching.
Thank you for that very informative presentation. It's now time for our question and answer session. So if you do have any further questions, please do send those in. You can submit these by typing into the questions box on the right of your screen. So to begin with, we've had a question in asking about the details for case study 2. Dr. Wang, do we have you there?
Hi, hello.
Hi, Dr. Wang. Can you hear me all well?
Yes, I
Excellent.
can.
Sorry, off you go.
Yeah, I heard the question. So the details for case study 2 is CrossMab with CH1CL crossover, and it's actually it's a very typical bispecific antibody. So monovalent for each of the bispecificity. So after the first step purification using protein A chromatography, actually the purity, if you only look at the SDS page and SEC-HPLC, it's already perfect, like over 95%. However, so we have this recently optimized LC mass method. It can tell us further details of the pairing. So actually, based on the SDS page SEC-HPLC, you can't tell the mispairing of the light chain. As just now shared that the light chain can still have some mispairing issues, even with this CrossMab design, mainly because of the imbalanced expression level of these 2 light chains. However, with our optimized LC mass method, we can tell that there are indeed some mispaired light chain within the purified sample after the 1-step protein A. And we then, based on the identification of the mispaired light chain, we then chose to use capture select kappa, which is another affinity chromatography, to further remove the mispaired species. And in the end, after this second step purification, you see the purity is still over 95% based on SDS page and SEC-HPLC. Actually, from the LC mass, it tells you that the mispaired species were totally removed. Hope I've answered the question.
Great, thank you very much, Dr. Wang. So the next question we've had in for you is, so it looks like the yield of the antibody from the two case studies differ a lot. What is the major cause for the difference? Is it because of design or the antibody itself?
Well, so as you can see, well, indeed, there are different designs, right? The first case, Glofitamab, was using the VHVL crossover, while the second case was using CH1CL crossover. However, from our experience, the difference of the expression level or the difference of the final yield was mostly due to the titer of the antibody, the titer of the molecule itself is mostly sequence dependent. And of course, if you're not very experienced with the purification, you can actually lose a lot of the material during the purification process. However, if with optimized purification methods, you can get a high recovery throughout the purification. So for our experience, based on our experience, the final yield was mostly dependent on the initial titer of the expression.
Excellent, thank you for clarifying that for us. So we've had another question in here. Could you explain more about how you match the light chain better?
How we match the light chain better? Okay, so there are two different heavy chains and two different light chains, right? So you wanna pair let's say you wanna pair light chain 1 with heavy chain 1 and the light chain 2 with heavy chain 2. And if you did nothing, you just translate this, transfect these 4 different chains into the cells, you will have up to 16 different recombinations of these 4 chains, right? And the final yield of your desired format is only a little bit about 10%. But with the CrossMab technology, with this strategy, you can take there. So just now in the talk, I have introduced the 3 different strategies. One is Fab crossover. So you put the CH1, you put the VH and the CH1 domains as the light chain, and then you put the light chain, the corresponding light chain to the FC region, so as part of the heavy chain. And so in this way, the light chain can only pair with the crossed light chain, can only pair with the crossed heavy chain. And this is the Fab crossover. Similarly, for the VHVL, right, crossover, so the idea is after the crossover, it only paired with the crossover of the other chain. So hope I have explained this clearly. And otherwise, I believe that the slides I used for the talk will be shared later. You can have more details by yourself. So the slides show clearly how in a picture format, how the crossover will encourage the correct pairing.
Great, thank you very much, Dr. Wang. We've had a good few questions come in, so keep those coming in, guys. So your next question is, do you have any data regarding stable production of bispecific antibodies? And what are the most important points to consider in terms of ensuring appropriate expression levels of each chain? Or does CrossMab technology omit the need for optimizing expression levels?
First question, stable production. So we do have a stable cell pool generation of bispecific antibodies. And yeah, hope I've already answered this one. But of course, before generation of the stable cell line, so we do highly recommend to have a transient expression as a screening step. And for the second question, ensuring appropriate expression levels. Yes, so actually even with the transient expression, we would normally recommend an initial transfection ratio screening of before a larger scale production. The reason is to get a balanced expression level of each of these 4 chains. So this is very important for the correct association of these chains as well. Yeah, for sure, the answer is yes.
Okay, great, thank you. So we have another question here for you. What is the FDA approval level for aggregate? For bispecific antibodies? Is it similar to monoclonal antibodies?
So FDA approval level. I'm afraid I don't have the exact number, but based on our clients' requests, normally they will require a higher than 95% SEC-HPLC purity. So which means less than 5% of the high molecular weight aggregates.
Okay. So we have, the next question we've got in here for you is, given these similarities in size and physiochemical properties between 3/4 and full antibodies, what specific assays are typically deemed acceptable within the industry for assessing yield and purity after each purification step? E.g. purity by SEC HPLC can account for both 3/4 and full molecule. It's quite a long question there for you. So.
Yeah. So just give me a few seconds to digest first.
Yeah, definitely fine.
Given the similarities. By SEC-HPLC, actually, SEC-HPLC, it's very hard for SEC-HPLC to tell a full-length antibody from a 3/4 antibody. That is, which means a loss of one light chain. So normally we will tell the amount, the presence and amount of the 3/4 antibody based on SDS page or CSDS and LC mass. Yeah, maybe on SEC-HPLC, you can slightly, given a very high resolution, you can slightly see a small shoulder, but it's not like a definite peak from the full-length antibody. So it's you have to rely on a combination of different QC tests.
Great, thank you for clarifying that for us. So your next question is, what are the routine assays used to determine yield and purity?
Well, as shared just now, okay, this question is from the same person. Hi, Nata. So the most commonly used QC tests are first SDS page, or alternatively CSDS, and SEC-HPLC, and LC mass. For LC mass, we have different like a finer tests like deglycosylated mass and reduced mass, intact mass, deglycosylated and reduced mass. So quite a few choices. And of course, we have some special tests as well in terms of HPLC. So we can also offer a reverse phase HPLC, yeah, that can also tell you a lot of information, especially when you are working with a bispecific antibody.
Excellent. Thank you, Dr. Wang. We had another question in here. So the question is, they would like to find an effective vaccine against the rabies virus. How could bispecific antibodies assist in this?
Rabies virus, how could bispecific antibody? Okay, so, well, you can find a bispecific, you can develop a bispecific antibody that can target two different molecules of rabies virus, and you can also like let the bispecificity target on the same molecule but on different regions, so that it can resist to the mutants of the viruses.
Excellent. Thank you, Dr. Wang. So we've had a few more questions come in here. Will antibody internalization be an issue for the therapeutic efficiency of the bispecific antibodies, and how will the community deal with that?
Internalization. That's a good question. So from my understanding, if the antibody, if you do some special engineering on the Fc region, so that it has a higher binding affinity to the neonatal Fc gamma receptor, actually after internalization, your therapeutic antibody can be recycled and reused. It can have longer efficiency. It can have longer time in the human bodies or in the target. But I'm not sure it's a good thing or a bad thing. So it can actually extend the effectivity of the antibody, but on the same time, it can also lead to some toxicity, some side effect as well. So it's really like it depends how the actual mechanism of the therapeutic antibodies it does inside of the body.
Great, thank you, Dr. Wang. So the next question we have for you here is, so you mentioned that one can lose a significant amount of bispecific antibodies during purification. Do the general purification methods, such as using protein A, not work as well for bispecific antibodies as they do for regular monoclonal antibodies?
Oh, actually, protein A is a very good method. It also applies to bispecific antibody. Normally, most commonly, the first step, the catching step, is done by protein A affinity chromatography. But unlike monoclonal antibody, so normally it requires a bispecific antibody requires more than one step protein A. So after protein A, we normally do some QC tests to verify the purity of the antibody. And based on the QC results, we will then decide on the method of the following steps. So the short answer is protein A applies to the purification of bispecific antibody very well.
Great, thank you very much. So this next question is, how can your team further improve the poor expressed antibodies?
Well, if there's enough resources, we can even do a DOE focusing on the upstream optimization. So for example, different expression platforms, we have different cell lines, and we have different codon optimization methods. We have different choices of a signal peptide, as well as plasmid ratio and expression time. What we have, so like you can name it, we have all different parameters to consider to optimize on.
Excellent. Thank you for clarifying that for us. So the next question we've had in is, what is a typical titer that GenScript can provide for CrossMab bispecific antibodies?
Well, as you can see, the highest titer. So as you can see from the two cases I shared, so one can be like a few hundreds milligram per liter of cell culture, but sometimes it's really, as mentioned already, it's really sequence dependent. Some sequences are low expressors, and it can be as low as below 100 milligram per liter. So, but still we have ways to optimize on the improvement of the titer.
Great, thank you, Dr. Wang. So this next question is, have you done stability/developability studies on both CrossMabs?
On both CrossMabs, I guess both CrossMabs, you mean the two CrossMabs I shared just now in the talk, right? For the stability, so we do have stability tests like a freeze and a thaw, and heat stability test as well. Developability, so we don't have a general developability studies, unfortunately.
Okay, thank you. So your next question is, can we create bispecific antibodies if we only know the two target proteins?
If we only know the two target proteins, I think the first step has to do some antibody discovery work, like so through immunization on mouse or rabbit, or doing some fish display or some kind of display technologies to get the antibody against each of the target protein first, and then with the variable region of the proteins of the antibodies, then you can construct into a bispecific antibody like a CrossMab or other format.
Excellent, thank you for clarifying that. So we have another question here. How long is the turnaround time regarding from the design, testing, optimization, and then to having the final antibody for customer to test?
If it works smoothly, it can be as short as 2 weeks. But if it involves some like plasmid screening or even some upstream screening, it can take longer.
Okay. Excellent. So our next question is it typical to use 2 steps for affinity purification for CrossMabs?
Normally 1-step purification is not enough for a CrossMab, unless you are really, really lucky. But we do have some good experience with the production of Glofitamab, since it's already a commercialized antibody. So it has been optimized for like it have gone through quite a lot of optimization. But generally speaking, a bispecific antibody requires more than 1 step, at least 2 steps. Sometimes it can take 3 steps for a desired purity. And it's not limited to affinity method. It can, so the second step can be SEC, which is size exclusion chromatography. It can also be ion exchange, and it can be a multimodal chromatography as well. Yeah. So it really like a case by case.
I've just noted that I think it's from the same person. We've had the question. I think they're referring to affinity resin. I'm not sure if that changes the answer slightly.
Yeah, still the same.
Okay, cool. Just double checking. So your next question, Dr. Wang, is they're wondering if reverse phased high pressure liquid chromatography normally involves an organic solvent. What would that affect the stability of the bispecific antibodies during the course of the analysis?
Right, it can, but you will tell after examining the result of the HPLC. Yeah, I agree.
Excellent. Right, so we're going to wrap up there. I'd like to thank our expert speaker for the very informative presentation and a discussion. Oh, hang on, we've had some questions come in. We've got time for those. So,
Sure.
is the plasmid ratio important for the success of CrossMab production?
Oh, yeah, of course, definitely. So as shared just now, the reason doing a plasmid ratio screening is to get a balanced expression level of each of these 4 chains. So yeah, definitely plasmid ratio is very important.
Great, thank you very much. So yeah, we're going to wrap up there. I'd like to thank Dr. Wang for a very informative presentation and discussion, and a big thank you to everyone for joining us online. We hope you found this a worthwhile session. If you do have any further questions, please feel free to email me at editor@selectscience.net, and I will follow up with your questions for our speaker. Remember, you can download related resources in the tab to the left of your screen, and this will include the certificate of attendance. And if you'd like to listen again to today's presentation or invite a friend to listen, it will be available to watch on demand in just a few days. Goodbye, and thank you once again for joining us.
Thank you.